typically. There are two types of molecular assays. The first is what we hear on television often, PCR tests. These are the most common and are manufactured by very large companies like Roche, who appear to be the market leaders right now in the US, but followed closely, though, by three local San Diego companies, Hologic, Dell. These type of assays are thought to detect the virus earlier in a patient's infection, so that's an advantage, but also they do take the longest to perform and are at the fewest number of sites in the US. Isothermal molecular assays like Abbott's ID now, and I know that everybody who watches the news has seen the Abbott ID now at this point, but that particular assay doesn't isolate and purify RNA, but it is faster and available at more locations, which can be useful. But as reported in the press, they don't detect the virus until a few days after you would see it in a PCR test. So in other words, they don't detect it till a few days later in the infection. Both technologies are very good depending on the use case. And in other words, how they're actually used. PCR tests are thought to be best in diagnosing patients with symptoms in the acute care setting. Point of care assays like Abbott's ID now are more useful in the outpatient setting. The second category, which is what we're becoming somewhat known for, is the rapid antigen assay for SARS. These are tests that detect one of the four proteins that is highly specific to the virus from nasal swabs. Quidel's rapid antigen assay runs on a small fluorescent immunoassay analyzer called Sophia. There are 37,000 Sophia analyzers in the US currently, which until recently were more commonly known and used for influenza testing. But because of the demand for SARS testing, we're now shipping more Sophias monthly to urgent care centers, retail clinics, pharmacies, physician offices, and hospital networks as well. Sophia SARS, which is the only FDA EUA cleared rapid antigen assay available today in the US, appears to detect the SARS virus as early in the infection as the rapid isothermal assays, but not as early as PCR. On the other hand, the assays are fast, economical, very easy to use, and with 60,000 analyzers dispersed across that number of sites throughout the US as we approach the next respiratory season, very well suited for the frequency of testing that's going to be required, particularly as we consider what to do to get people back to work. Our current capacity to manufacture these assays is up to 7 million tests per month, and by as early as the first quarter of next year, we will add another 5 million tests per month, so a total of 12 million tests per month next year. About 1/3 of our tests so far have been performed in the urgent care setting, and then in that environment, positivity rate surprisingly has been 9%, which is a bit higher than people had thought, I think. And then the third category, serology assays, are effectively the reverse of antigen assays and are tests that detect the patient's antibodies to the SARS virus, which would indicate that a patient had been exposed to the virus and had generated an immune response. Since we're looking for antibodies circulating in a patient's blood, the sample is either plasma or serum from a venous draw, or in the case of the Sophia serology assay, a whole blood finger stick. Serology assays, whether run in centralized lab settings or at the point of care, can be useful, but with a couple caveats. The first is that when prevalence is low, the positive predictive value is probably not good enough. In other words, when prevalence is low, positive is too often a false positive, even in very good performing assays. And second, not enough is really known about whether antibodies to the different viral proteins confer immunity. For example, you may have antibodies to the nucleoprotein, so you've been exposed. But to be immune, do you need antibodies to one of the two spike proteins or both? And how long does immunity actually last? So in summary, serology, the third technology that we're talking about, these assays are likely to be useful. How useful is yet to be determined and defined.
Thank you so much, Doug, for explaining how the different tests work and the pros and cons of it. Actually, I wanted to call on Joshua Trotta from tests. How does the reliability compare to other things out there on the market? What are your differentiation? And as well as what does Thermo Fisher have planned for the future as far as future testing?
Sure. So thanks, Pam. I appreciate it. And hello to everyone this afternoon. So yeah, from a Thermo Fisher scientific perspective, the solutions that we're providing predominantly in the market today have been on the PCR side. So as Doug was just alluding to, they're PCR-based testing. The solutions that we have currently, when you look at assays, what are some of the things that differentiate assays in the market? So within Thermo Fisher's solution, it is a multiplex assay. So we actually target 3 areas. So the ORF1A, AB gene, the S protein gene, and the N protein genes. With that, then, as far as making the, I think the ability for having a multiplex assays just allows people to increase their throughput. So as we saw, I think early in the process of needing more testing to be done, having simplified solutions that a multiplex allows to have as opposed to individual tube assays just allows people to get more coverage across the assay. Within our solution as well, we include both positive controls as well as some of our extraction controls, just from a security perspective and the safety for those that are actually running the testing. So it's just adding some of those added benefits to users downstream, and then inclusion of things like interpretive software. I think some folks probably know Thermo Fisher and some of the brands of Applied Biosystems. So an added advantage is that we are an integrated supplier. So our solutions have been approved on 9 different configurations of our platforms. So when we look at the use of the solutions, it's pretty broad across the different types of samples that can be run, as well as the instrument configurations that just allows people to have the support kind of on a global level that they need. So really, I guess when you get down to the limit of detection within our IFUs, it's 10 copies per reaction. And then as you look at just the really the analytical performance or the clinical performance of the assays, we've really done comparisons. I think we have data against both the Roche solutions as well as the CDC assay that show at least 95% concordance. When you take that, then you can say, well, what about the non-concordant results? We then use sequencing to look at those results as well, and just to show that indeed that they are positive results. So it's only as good as data that's available to show concordance. So those are kind of the 2 that we've actually used. So that's just from a deployment. You asked about a little bit, Pam, of other things. So I think you've probably seen, a few folks have seen within the marketplace, Thermo Fisher has also done some development work with Wuxi AppTech as well as the Mayo Clinic. So looking at additional testing as well as supporting vaccine developments. So Thermo Fisher is broadly involved in the manufacturing side of enabling customers to actually develop tools, both on the serology side as well as the vaccine side, and then looking at kind of the next iterations of where do you go with testing? Is it just going to be in the the centralized testing environments, or is there a need for like higher throughput testing? So we are looking at other areas to kind of support customers on a global level for higher throughput testing needs.
Thank you so much for all that information. A lot going on with Thermo Fisher for sure. Not just in testing kits, but in supplying supplies to the companies doing the research.
Yep.
So thank you. Maurice, Next, I want to call on you to tell us more about your technology related to the Panther Fusion SARS-CoV-2 testing that you do, which has the ability to make millions of tests and run them for millions of tests per month. Can you tell us more about that technology as well as innovation that you have planned?
Yeah, sure. Now, our technology and what sort of sets it apart, I think, is mostly in the automation and the unique chemistry that we use. But actually, we have two systems on the market right now. The first one that we launched was with what was called our Panther Fusion technology. And so this has what we call a fusion module that's added onto our standard Panther instrument. And that provides us with a capability to run different chemistry, and it gives us also what we call our open access feature, which allows us to very rapidly develop new tests to put onto that system without having to do a lot of different major software changes to the whole larger system. So that was the first assay we launched, but quite quickly we realized that with 100 systems in the market, the demand was really outstripping what we had there. So we actually turned to develop a test on our Panther system, which is our basic instrument, which we have thousands in the field right now, over 1,000 in the US. And so with respect to technology here, they both have the capability to do full automation such that it's as easy as taking a specimen, wherever you collect that specimen, and gets sent into the laboratory. You just take that specimen, put it directly on the instrument and press go. And so it's completely automated from start to finish, and we can do over 1,000 samples a day on that. So really one person could run 1,000 samples per day on an instrument. In addition to the automation, which makes it easy to run, for example, our Panther has a unique chemistry, which we call our transcription-mediated amplification, or TMA. And this has been known historically to be exquisitely sensitive. So that gives us great sensitivity in the product. And when we combine that with our target capture chemistry, which allows us to pull the virus or the viral nucleic acids out of the sample and essentially concentrate it, combining those together gives us a very, very sensitive assay and a fully automated system. So that's sort of what is unique and separates it from some of the other things you'll see out there. And it allows you to put the instruments in a small laboratory, small hospital, large hospital, or the throughput capability allows you to put this in the largest labs in the country, the largest reference labs as well. And I think because of that flexibility, I think what you'll see is that the majority of molecular tests being run in the US right now are being run on the Panther systems. For innovations going forward, it's interesting, but most of our innovation is looking at scale up of capacity, because it's just, we've never ever seen this kind of volume. We probably do the largest share of testing for sexually transmitted infections in the US and probably across the world from a molecular standpoint. And those are, millions of tests a year, tens of millions of tests a year. But even having capacity to do that, we're seeing that quickly outstripped by the demand for COVID-19 testing. So we've got our R&D scientists working with our engineers and our manufacturing teams to do novel ways to create new ways to get products out the door, build more tubes, build more swabs, and more chemicals to do everything. So that's where we put a lot of effort. Actually, we have done additions to the products that we have on the market, different claims, different specimen types that we can now do, lower respiratory specimens, even looking at other things like at-home collection and less invasive collection. We've originally went with the standard nasopharyngeal swabs, which are difficult. You need a healthcare provider to collect those, but now we've gone to nasal swabs, which anybody can collect, and now that's what you're seeing in the drive-through clinics. It makes it much easier to do that. So scaling up and adding different capabilities for indications has really been what we're looking at now to expand our abilities to do more testing.
Thank you so much, Maurice, for that information. And next I wanted to talk to Susan Tusi from Aluminum, and I'm really excited to have her here. I found out about the news related to their recent press release, and so asked her to join us on Monday. And they have the first FDA use authorization for sequence-based COVID-19 diagnostic testing. And so, Susan, it would be great if you can elaborate on that.
It's great. Thank you, Pam, for the opportunity, and nice to see all the panelists and hear from them. I guess the approval of our test is so new. The diagnostic options that exist for COVID. We are the first diagnostic NGS-based test that has received emergency use authorization approval from the FDA. So our test, in addition to being very high scale, it is a complete sample to answer workflow. It uses currently upper respiratory specimens, including nasopharyngeal and oropharyngeal swabs, although, we are working on an amendment that would add other, maybe easier to collect sample types, such as a nasal swab or saliva. The test runs in 24 hours end-to-end using our NextSeq 6000 sequencing system, our high throughput sequencing system. It allows for 3,072 samples run of a NovaSeq. And it actually, if you're automating the upfront sample preparation, you can run the NovaSeq twice in a day and get up to 43,000 samples in a week. So it is a very high scale kind of sample output that we're adding kind of into this fight for COVID. Additionally, there's more information that comes of using our sequencing technology, and the test includes kind of 98 amplicons that cover the entire sequence of the virus, in addition to 11 human RNA controls for every sample reaction. So we do believe in controls and QC validation, of course. We, not only does the test allow you to have kind of a yes/no detection in a clinical report, but also allows you, if there's enough, the entire viral genome sequence, which gives you information into the function and kind of biology of what's going on, and being able to track and change over time. We allow for then submitting that easily to GISAID or other public sources for sharing and future surveillance. So we believe it's an important kind of element to add into this fight against this pandemic. Additionally, we intend to continue to develop out the test onto our benchtop platforms, onto our NextSeq platform, to allow for wider distribution. We have a large fleet of all of these instruments out in the field, and so it allows for scaling of COVID testing. We also have released a respiratory virus panel, which and many of our customers have widely adopted at this point, and this allows for not just detection of the current virus, but over 40 other pathogens that can be detected. And we believe in the fall, as people come back and they're worried about influenza, other mutations of this virus, other viruses that could be present, other pathogens, kind of co-infections, and also the antimicrobial kind of resistance markers. These are all elements that will be important for preserving human health. And so these are other areas that we're invested in. Additionally, made a toolkit available in our base-based sequencing hub, our cloud-based data analysis application. And there, we allow customers not only to upload these viral genomes, but also to import data coming from publicly available sources. You can use our analysis apps to then evaluate that data, to share it, and to submit additional viral into the community databases. We've made that function available for free for research use, and that will extend through October, so that our customers don't have to be concerned about the analysis time, compute and storage time of these applications. And so far, we've had more than 3,500 samples processed through our Dragon Analysis app, so we know that they're getting good uptake in the market. And we intend to continue to develop out the respiratory virus panels, extend our EUA, as I mentioned, to other easier sample types to collect and to self-collect. And you know, we believe that this, not only the scale of what we're adding into testing, but also the fact that, you know, largely other than the front end and the, you know, the swabs, and it's amazing the things that we're short of as a nation, as a global population. But other than, you know, the limited availability of those things, largely kind of our reagents and our instrumentation doesn't share the same supply chain as some of the PCR tests and other tests that we've talked about. So we also believe that we're, you know, adding just supply capacity into this testing fight that we're all in.
Thank you so much, Susan, for that great information. Speaking of supply chain, I wanted to call on Doug because as we go more and more opening up the world and having to do more and more testing to get our offices back up and running and to more of a place of normal, I was wondering if you could talk about the supply chain issues that we've seen, your insights, et cetera, and how you see things heading forward. Oops, Doug, your mic's off. You got to turn on your mic. Thank you.
Excellent. Thank you. This is the first time in my 35 years in this industry that demand wasn't a factor at all. And so I was just on the phone with a major reference lab a couple hours ago, and you know that everybody's spending a high percentage of the time talking about supply chain. We're actually right now building 4 more manufacturing lines 7, 8, 9, and 10. Each of those manufacturing lines will do about 30 million tests per year. So we, as we go into next year, should have capacity 240 million tests per year, or about 12 million tests per month. But supply chain is an issue. Even though I can build all that within my current footprint, even though I can develop the plant and the equipment necessary to do all of that, there are 8 key items in the bill of materials that we manage very, very closely. And a key lesson that we've learned was that while we do manufacture our own antibodies, for example, and other key elements of the assays, we were definitely too reliant on other companies to provide what we need for the less proprietary elements of the product. And often and we're getting those from often the same companies that my colleagues and other companies were relying on as well. I think everybody now knows that Puritan makes swabs, but originally, you know, they were shipping us 4 million swabs a month, and then they became famous, and we struggled to get what we needed to ramp up. So obviously, we're going to be in the swab manufacturing business here shortly as well. But what we did learn in this process is, in order to really have surge capability, we needed to move to more vertically integrated processes. So vertical integration is a key element to ensure that we control our ability to ramp up and down more quickly. And obviously, this is more costly. Many folks in my industry spend a lot of time getting cents per test out of their assays, getting their costs down, but clearly that was probably a mistake. And our ability to scale quickly is more important, and clearly a competence that all of the manufacturers in our industry need to rely on. So
thank you.
I'm a supply chain.
Yeah. Thank you so much. Just a lot of intricacies with that, and thank you very much. And Joshua, I wanted to ask you this question, especially as we're going out into the workforce and opening up offices as well. One of the questions that came in from the audience is related to the false positives and false negatives. And so, you know, let's talk about how we ensure that it's truly safe to go back to work and controlling those anomalies that happen with testing.
Yeah, well, I think it's important to note that all tests are not created equal. So there is different, as I mentioned, I think at the beginning of even amongst this panel, right, there's kind of different strategies on how companies have actually designed assays. But I think as part of the diligence of the global companies that are represented here, I think it's our responsibilities and our duties to actually, make sure that we have both robust analytically and clinically sensitive tests. I know obviously this is the balance between the regulatory compliance of trying to get tests out there and meeting needs versus actually having tests that are robust and people can stand by. So I think it's important as you look at the false negatives and false positives, to do the diligence on the tests themselves to see the performance. I think the last time I checked, there's something like 165 different tests that are out there globally into the market. When you look at some of the databases like Find, so there's a lot of tests, a lot of different solutions that are out there. I think I will say from a Thermo Fisher perspective, we're making 10 million tests a week, and we're scaling from there. So from a global supply, there are a lot of people using our tests. So from a robustness perspective, feel very good and supporting kind of the needs that are in the marketplace, and to have robust solutions that can be deployed globally.
Okay.
Didn't quite answer your solution because it's a broad question, and that would have to get into very specifics on specific tests.
Yeah, no, I appreciate it. And you know what? The other thing I didn't understand,
if I can add,
has a COVID assay that has been used with Rutgers for saliva tests. I was just wondering, from either you, Joshua, or anybody else in the audience, is there any plans to do saliva testing in the group? Because that's less intrusive as far as testing.
Yeah. So it's being used, I think you mentioned, on Rutgers.
We're looking at it.
Yeah. So yeah, Rutgers is currently using it as an LDT, but it is a piece for expanding submission under our EUA filings and CE-IVD.
Yeah, and whether it's saliva or a nasal swab, those are things that you could be collecting by yourself at home, sending in a sample, both pretty non-invasive that you could, a different testing paradigm, basically.
Okay.
Yeah, we see the same, and the sensitivity is not quite as good as the OP swabs, but we're working on it because there is just such a benefit to not having a clinician having to do the test.
Okay, thank you. And then Susan, really what we've seen with COVID-19 is collaboration is very important, and I know Illumina has been on the cutting edge of doing collaborations with working with viral function, disease transmissions, infection risk, as well as outcomes. Can you tell us more about what Illumina is offering with other places on collaborations?
Absolutely, and as mentioned, having the viral genome, having the sequence of the viral genome has been incredibly important in this fight. In fact, the first time the virus was sequenced and then off an Illumina instrument and made available to the global community, that really kind of started the clock on everyone's understanding and the research that was happening, and hopefully the acceleration of vaccine development. So it's incredibly important, and we've seen more than ever that we are very connected globally. And there's no such thing as this virus happened in this one region and it will stay there. And so being able to kind of share data is really important to this fight, and to future, as I mentioned, future mutations of this virus and other viruses that are going to plague mankind. And having the information, having the sharing and surveillance to respond sooner is going to make all the difference. I mentioned the toolkit that we've made available, and the fact that we've seen thousands of users already run samples in our toolkit. The fact that we're seeing, submission to GIS8 and NCBI and other data sources that are community led. I think there has to be a technology push on surveillance systems to make them, much more, I think, modern and connected globally. I think that's going to take some, kind of government push. I think it's going to take some push from private industry as well. But, the only way that we're going to actually be able to respond from the first time that we see something that's novel, like we are with COVID-19, the first time that we can respond, as well as, start the kind of research engine in the fight against it, I think it will be determined by, how connected we can make these data sources. We're absolutely invested in that. We think kind of having data sharing is incredibly important to the point that we're offering it for free, all the analysis for free, the sharing of data for free, and we intend to continue to push the technology side. We're very invested in our cloud software platform in really making the tools more available and more powerful globally to be able to have that sharing that stops the next pandemic.
Thank you so much, Susan. And Prasun, I know you weren't technically on the panel, but I wanted to just give you just a minute or two to quickly talk about your hackathon that you're running as we're talking about data sharing and collaboration.
Yeah, so I think we are running so American Association for Precision Medicine has a ACT task force. So it's abbreviated AAPM Coronavirus Task Force, and ACT Task Force is basically a global task force. So we have over 200 folks that are part of this task force. And what one initiative that we have is we thought to kind of build a community and foster a collaborative effort to identify specific problems that we are having as businesses are opening, as well as overarching, as you have seen from the previous panel, that the COVID-19 is going to stay. So then we have hosted this hackathon, which is at the end of this month. It's called Act to Hack COVID. So it's basically we are all coming together to find innovative solutions for the COVID-19 pandemic. So there will be genomics problems to solve. There will be supply chain problems to solve. There will be just overall data building, and as well as data analytics problems to solve. So yeah, so this will be a fun event. So I think we are looking forward to it, and I welcome you all to participate. It's Act to Hack COVID. That's if you search hashtag Act to Hack COVID. Yeah, you will find it. Thank you.
Awesome. Thank you, Prasun. And then Maurice, I wanted to ask you about how it's been working with the FDA, with them really giving a lot of emergency use access, et cetera, and how it's been, and also what advice can you give to other diagnostic companies that as they work with the FDA on COVID?
Yeah, sure. And I mean, from our experience, the FDA has been incredibly flexible. It helps, it certainly helps to have experience working with the FDA, and we've been fortunate enough over the past few years to have quite a number of products coming through the process. So we have a very good rapport with them and really a good understanding of how the FDA works. But outside of that, even if you've never put a product through, it's very simple to just look at what the FDA is saying. They put out a number of different guidances on how to get an assay through the process, the number of public comments and discussions that they've had. So a lot of it is, pay attention to what they say, pay attention to what your, competitor companies are doing. Anybody who gets a product through the FDA has gone through that vetting process and has done what the FDA has wanted them to do. So if you model your products based on what's been done in the past, that's also very useful. But one thing I'd really want to stress, though, and I guess I'd think of it this way. So a lot of people go into the healthcare industry because they want to make a difference. They want to help people. And through this pandemic, I know our teams here have been working 24/7, all around the clock from all different functions, trying to get products out the door. And the interesting thing is the FDA has been there with us. I recall through weekends, one Sunday we had 4 or 5 different interactions back and forth. So just like we want to make a difference in the world, so do they. They want to help to get the crisis out of the way as much as we do. So it's really about thinking of them as a helper and a friend versus a foe really here. They do want to help. Reach out to talk to them, ask them questions, set up meetings, educate them on your technologies. they really are out there to help you and not just to block your way through. So just having that attitude of thinking of it that way, developing those relationships with them, it pays off great dividends, and just understanding how they think, why they're thinking as they do. They've got a job to do, obviously, to keep our products safe. And so, yes, that might mean sometimes more work for us to do to prove how our products work, but it's all for the good in the end. And it's rare that you get something from the FDA that, that you would really think is, well, that just doesn't, that's not scientific medical sense. It doesn't make any sense. There's always a sort of a method to the madness there, and kind of seeing how they see things, understanding that, and like I said, treating them as a partner more so. We found that to be very helpful for us.
Thank you very much, Maurice. So we have 5 minutes left, and just wanted to hear from the group. And it's a very open-ended question, but wanted to hear everybody's thoughts on it. What can we do in the future to better prepare for a future pandemic, and what have we learned about this? And curious on how it's impacted you personally or your company. If everybody could give a brief summary. Maurice, since you're off, Mike, we'll go with you first.
Sure. I think for us, supply chain realizations has been a big one. Like I said, we do very, very high volume testing, and yet it was still overwhelmed. And so really understanding what that capacity should be or needs to be is really a big deal for us. And I think that's one of the things that slowed down some of the testing from our end. It's interesting, though, I think from a lot of ways with the partnership with the FDA, partnership with government, things have gone pretty fast from a manufacturer standpoint. I mean, we normally do not get tests out in a matter of a month or two. And so that's been actually impressive to see how fast manufacturers can actually work and get through this with the FDA. So I think it's actually been very successful to that end, but the supply chain has been difficult. But with a pandemic like this, as I was sort of alluding to earlier, everybody gets on board in the company. And like I said, nobody was frowning coming to work and working as hard as they were doing, because people really want to make a difference. And that's been the experience at our company, is so many people wanted to pitch in and help. And it's, people working together like we never have before. So it's really brought us together in some ways. Okay, perfect. Thank you. Doug, it looks like you have your mic off. Do you want to give a quick summary of your thoughts?
Sure. First, I would agree with Maurice. Tim Stenzel and the guys, his team at the FDA, have been fantastic. So when you look at future pandemics, epidemics, I don't think anything they've done really could have been easier. I would say that in preparing for the next pandemic, we've got to get better at sample collection, and it's the sample collection that actually created for most companies the greater delay, supply chain issues notwithstanding. But when we went into university systems to try to get samples that had been banked, we struggled, and we weren't the only company. It just turns out that the research deans at these universities are highly reluctant to give up samples. And in these settings, a lot of the patients, the samples are there and they're being withheld. And so I think as a country, it's not something we can probably solve as companies, but as a country, we need to figure out a better sample acquisition strategy so that lots of these companies can get access to the samples, because there's a lot of development talent out there, and there's a lot of companies like Hologic and others quite nicely, but still have to do the clinical work. You still have to show that the product works well, and despite the ease of use with the FDA, they need to see the data. So you need to have the samples in order to generate the data. So that would be my answer to how we could do this better with the next virus that comes around.
Thank you so much, Doug. Susan, can we hear from you on your insights? Oops. Sorry. Yeah.
I have been impressed with kind of the response of, all of my co-panelists here in terms of scaling up testing and so forth. I guess my view, I would like to be on the kind of preventative and how could we have slowed down or stopped the spread of this much sooner. And so that, the thoughts on data sharing from the first time that we had the viral sequence of COVID and knew that it was a novel virus, knew that, the early death rates that we were seeing are much higher than what we'd seen before. Had we shut down borders earlier, had we done social distancing earlier, had we really been able to respond because of, surveillance being in place and us really understanding that this was something new and that we need to take action earlier. I think that's where we can make the biggest difference in the future. And so really taking, I think the technology exists, we're able to a lot better. And then, beyond that, I think scaling up testing and being able to kind of precision isolate people and, reduce the amount of transmission. I think all those things are incredibly important. And hopefully, this engine that we've cranked up will continue to improve. And I would just second like what we've seen from the FDA response. I mean, internally at Illumina, 50 days ago, we didn't have this on our radar as a project, and here we are with, an EUA file. So, people are incredibly passionate. They want to make a difference. All of us and our companies want our technology to work. It's great to see how hard the FDA works every weekend with us as well. So I think, I'd push more on the preventative side than on the responsive side at this point.
Awesome. Thank you so much, Susan. And Joshua, do you want to give some remarks as well?
Yeah. So thank you. I think just adding on to what was already been said. When I think about on the response side of things, I think there's some lessons in historically in the engagement of this group that is here. You see that we are all from commercial companies, and that is not generally the first place that the government would historically look to go to say, hey, let's tap into this broad swath of science and of scalability, which I think there's some learning in that process of that. Equally, hopefully, we're in the I think on the global pandemic role, the impact of what companies that are commercial suppliers actually have played, and the more that we can actually get there faster to say there are different pieces that we do well, which is scalability and manufacturability. Although we have constraints and there have been challenges that people have talked about, the ability to scale to the millions and millions of tests that are actually needed, I think is an important one, and hopefully one that is not soon forgotten of how we collectively, I think both across the private and public sectors can actually really work well together. And I think to that point, one thing, and just on a personal level, you had thrown out there, I think, I've been able to really engage with a lot of our global counterparts and the World Health communities. So with UNICEF and WHO and others, where there's a lot of testing even on a global level outside of the United States. There's lots of low and middle income countries that have struggled to find access to testing. And I've been very thankful to be part of an organization to really sponsor and support those countries as well, to be able to provide solutions that can help some of these areas that really can't help themselves. So I've been really thankful to have that kind of passion from the Thermo Fisher leadership team, just to be able to enable solutions into some of these low and middle income countries as well.
Thank you so much, Joshua. And I really want to thank all of our panelists for their participation. And once I turn off the presentation, everybody's going to be sent back to the exhibit hall. And for the next hour, I encourage everybody, our attendees and speakers, if they can stay, to actually network with everybody. And you just tap on the table that you want to see. And there, in addition to where the vendor tables are, off to the side, there are banner ads where you can see videos about the companies. So just been such a pleasure, and I'm so happy that this all came together, and really appreciate everybody being here. Thank you so much.
Thanks.
Thank you for putting it together.
Thank you so much. Nice to meet all of you.
Thank you. Great to see you.
Bye-bye.